In the lysogenic cycle, the virus's genetic material will make its way inside the nucleus of the B cell. Upon entering, it will integrate a part of its own DNA within the cell's DNA. The seemingly healthy B cell will continue its normal functions. This dormancy of viral DNA is called latency. EBV is essentially using the B cell as a hiding place. Note that lymphocytes only multiply in certain circumstances, mainly when they're activated by the antigen their receptors are programmed to recognize. This is called cloning. If a B cell clones itself with EBV DNA integrated within its own DNA, each one of the clones will also contain viral DNA. With EBV invasion of B cells, the lytic cycle always follows the lysogenic cycle. In this cycle, something will trigger the viral DNA to end latency and begin the process of synthesizing viral proteins. There are many theories as to what these triggers are, but no one knows for sure. mRNAs will decode the viral DNA sequence and send the message to the B cell's ribosomes. Synthesis of viral proteins will commence. These proteins will form virions, assemble in large quantities, and exit the B cell, sometimes causing lysis. The new viruses will either search for another B cell to infect or an epithelial cell. If the viruses infect an epithelial cell, it will initiate the lytic cycle. Infection of epithelial cells can lead to infectious mononucleosis. EBV can actually live quite peacefully inside humans with little to no impact on homeostasis. As stated before, 95% of the worldwide adult population actually has the virus inside of them. However, problems can arise in the event of replication and large-scale lysis. If the already weakened immune system can't control replication, a pathology may follow. Each year, almost 200,000 people will become ill with an EBV-related disease. There appears to be a link between EBV and some cancers. For example, almost all cancer cells found in anaplastic nasopharyngeal carcinomic tumors contain EBV DNA. Of the individuals who have recently contracted EBV, 30 to 50% of them will develop infectious mononucleosis, usually within 2 months. Those with infectious mononucleosis are typically symptomatic for about a week, exhibiting a fever, lymphadenopathy, and pharyngitis. However, EBV can spread to others even when asymptomatic. Roughly 5% of infectious mononucleosis cases can develop into more serious pathologies, such as lymphocytic meningitis, encephalitis, encephalomyelitis, polyneuritis, mononeuritis, and Guillain-Barré syndrome.